Hello everyone, welcome to my channel Anupma Biology Classes. This is the sixth lecture in the series of cell biology. In this video, our topic is cell cycle and cell division. This is the last video of the cell biology series. After this video, we will start reproduction in organism. This video is from CBSE class 11th topic. Now, the first topic of this video is cell cycle. You know that multicellular organisms start life as a single cell, which is a zygote. Zygote is fertilized egg, and one thing is in the chromosome or DNA of a parent cell must be doubled exactly if the daughter cells formed by its division, or to have identical information about their structure and functioning. So this emphasis on the unity of life. Cell cycle was described by Howard and Pele in 1953. It is the sequence of events by which a cell duplicates its genome and synthesizes other cell contents and eventually divides into two daughter cells. During the cell cycle, period between the two successive cell divisions is called generation time. So this generation time depends on the type of cells and the external factors such as temperature and food and oxygen supplies. Like if you talk about bacterial cells, which divides every 20 minutes, epithelial cells of the intestinal lining every 8 to 10 hours, and onion root tip cells may take 20 hours for the generation. While the mammalian cells divides once in 24 hours and yeast cell only in about 90 minutes. Now, the phases of the cell cycle. It has two main phases in which the first one is a long undividing state that is known as interphase or intermitosis or I phase. And the second is a shorter phase of nuclear division which is known as mitotic or M phase. These phases are followed by a shorter phase of cytoplasmic divisions, that is cytokinesis or C phase. Here, the various phases of cycles are regulated by proteins, that is cyclins and cyclin-dependent protein kinases. In short, you can say CDKs. There are two checkpoints, first between G1 and S and the second in between G2 and M. So, in this picture, you can see all the phases of the cell cycle which has longer interphase which is divided into G1, S and G2 phases and the short mitotic phases. In G1 phase, cell growth occur. In S phase, synthesis of DNA occur. In G2, cell growth occur, and in mitotic phase, two daughter cells are formed by division. And here there are two regulatory proteins which check the process. First in between G1 and S and the second in between G2 and M. These checkpoints takes decision about the cell division. Here the first phase, that is the interphase of the cell cycle. It involves a series of changes that a newly formed cells and its nucleus undergo to become fit for division. It is a period of intense synthesis and growth. The interphase lasts for 10 to 20 hours and takes 75 to 90% of the entire generation time. It has three phases, in which the first one is G1 phase. It is a stage of initial growth of a newly formed cell. It is also known as gap 1. In this phase, cell grows, the cell synthesizes various enzymes and nutrients that are needed later on for the DNA replication and cell division. In this phase, there is a synthesis of biochemicals like RNA, proteins, enzymes for the DNA synthesis, amino acids for histone formations, and nucleotide and ATP formation occur in this phase. Second is S phase. It is a stage of replication of each chromosomes by synthesis of a new DNA molecule on the template of the existing DNA. It is the period of whole cell DNA synthesis during which the cell replicates its genetic content, a normal diploid somatic cell with a two-end complement of the DNA at the beginning of S phase acquires a 4n complement of DNA at its end. Now, the third phase is G2 phase. It is also known as gap 2. It is a stage of further growth of the cell and preparation for its division. This stage is most cells last for 2 to 5 hours. In this, the cell has grown, DNA has been replicated, and now the cell is almost ready to divide. The last stage is about prepping the cells for mitosis or meiosis. At the end of this gap, another control checkpoint to determine if the cell can now proceed to enter M and divide. Here, DNA repair. Repair of damaged DNA sequence also takes place in the interphase. So, an interphase is sometimes described as a resting cell, but it is not correct. In fact, interphase is the most active period of the cell, and by some workers it is described as energy phase. Now, this is the whole picture of the cell cycle with time of the cycle. Now, in first, that is the interphase, which has three phases. First one is the G1 phase, then the S phase and G2 phase. In G1 phase, cell metabolically active duplicates organelles and the cytosolic components start replicating centrosomes. In the S phase, DNA is replicated, and in the G2 phase, cell growth continues, enzymes and other proteins are synthesized, and replication of the centrosome is completed. After that, a mitotic phase, that is known as the M phase, in which there are many stages like the prophase, metaphase, anaphase and telophase. So in this stage, the division of cells form two daughter cells. If the daughter cell wants to enter into the cell cycle, it enters into the G1 phase, and if it doesn't want to enter, then it exits from the cell cycle, that is the non-dividing cell. And this cell cycle, when exit, it's known as G0 phase. So the G1 phase is for 8 to 10 hours, S phase for 6 to 8 hours, and the G2 phase for 4 to 6 hours. So this is the complete cell cycle. Now, the second phase of the cell cycle, that is the mitotic phase. It starts with the nuclear division corresponding to the separation of the daughter chromosomes and usually ends with the division of cytoplasm. It is aimed at orderly distribution of the already duplicated chromosomes among the two daughter nuclei. After mitosis, cytokinesis occurs, where separation of two daughter cells which are diploid and contain identical sets of chromosomes and exactly the same hereditary information as the parent cell. After the completion of the mitosis, the daughter cells may enter the G1 phase of the next cycle or the G0 phase. So, what is the G0 phase? The cells which are not to divide further do not proceed beyond the G1 phase and start undergoing differentiation into a specific type of cells called quiescent stage or G0 phase of the cell cycle to distinguish them from the G1 cells, which will soon enter S phase. The alternative sequence of G1, S, G2 and M phase is called the cell cycle. Here, the cell cycle and their checkpoints, which is on three positions in the cycle. So at first you can see in the picture the arrow indicates the checkpoints. First on the cell growth checkpoints, which occurs towards the end of growth phase 1 and checks whether the cell is big enough and has met the proper proteins for the synthesis phase. If not, the cell goes through a resting period until it is ready to divide. Second checkpoint is DNA synthesis checkpoint, which occur in the S phase. Here occurs during the synthesis phase and checks whether DNA has been replicated correctly. If so, the cell continues on to mitosis. And the third checkpoint is on the mitosis checkpoint, which occurs in the M phase and checks whether mitosis is completed. If so, the cell divides and then cycle repeats. So these are all the checkpoints in a cell cycle. Now, cell division. It is meant for the multiplication in unicellular organisms and in multicellular organisms. It brings about embryonic development and growth and also plays a role in repair and maintenance of the body and also in reproduction both asexual and sexual. Now, the modes of cell divisions. So, there are three main modes of the cell division like amitosis, mitosis and meiosis, in which the first one is amitosis. It was first discovered by Remak in 1841 in the RBCs of the chick embryo and later described by Fleming in 1882. In this picture, it is a case of amitosis where cell divides and form two same daughter cells. Now, if we talk about the mechanism of the mitosis, here it occurs without the formation of a spindle and appearance of chromosomes. The nuclear envelope remains intact. So, it is also known as direct division. The nucleus of a cell elongates and develops a constriction around its middle. The constriction gradually deepens and finally cuts the nucleus into two daughter nuclei, which you can see in the picture. This picture shows a mitosis where a parent cell has nucleus and cytoplasm. Now, the parent cell starts dividing at first karyokinesis, which means division of the nucleus, and then cytokinesis, which means division of cytoplasm. At the end of cytoplasmic division, constriction creates and the two daughter cells formed. So this is the complete process of the amitosis. A similar constriction appears in the cytoplasm between the two daughter nuclei and divides the parent cell into two daughter cells, each with a nucleus. And the daughter cells receive equal amount of nuclear and the cytoplasmic material. Now, where the amitosis takes place? So, the amitosis takes place in the mammalian cartilage, in the growing embryonic membrane of some vertebrates, in the degenerating cells of the hair plants, in the diseased tissues and in the old tissues. Now, the second cell division, that is the mitosis. It was described by Edward Strasburger in the plant cell in 1875 and the latter by the Walter Fleming in the animal cells in 1879 and term mitosis coined by Walter Fleming in 1882. It takes place in the somatic cells in the animals. Hence, it is also known as somatic division because it occurs in the gonads for the multiplication of undifferentiated germ cells. In plants, it takes place in meristematic tissues and during the growth of leaves, flowers and fruits. Mitosis lasts on an average from 30 minutes to 3 hours. It is the division of a parent cell into two identical daughter cells, each with a nucleus having the same amount of DNA, the same number and kind of chromosomes and the same hereditary instruction as the parent cell. So it is also known as equational division. Now the mechanism of mitosis. There is a two main events in the mitosis in which the first one is karyokinesis or duplication of the nucleus and the second is cytokinesis or the division of the cytoplasm. So mitosis involves a series of important changes in the nucleus as well as in the cytoplasm. Therefore, it is also known as indirect division. Now, the first main events in the karyokinesis, which is a continuous process, and here the four main phases occur in this karyokinesis, in which the first one is prophase. Prophase lasts for about 50 minutes. It is the longest and most complex phase of the cell division. It has three substages: early prophase, middle prophase and late prophase. It is the first phase of the mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. So, in the prophase, there are three substages. That is, early prophase, where the formation of esters and a spindle occur. Then, middle prophase, where the chromosomes become shortened and thick and chromatids uncoil. Nucleoli starts decreasing in size, and the nuclear envelope begins to break down and disperse into the cytoplasm. And the third is the late prophase, where the spindle assume in the proper form and size, nuclear envelope break down fully, centrioles pairs are pushed to the opposite ends of the cell by the growing spindle. The esters and spindles are together referred to as the mitotic apparatus. So, in this picture you see the chromosomes which attached with the centromere and the microtubules. This is the process how the chromosome segregates during the mitosis because ester rays and the spindle formation occur in this phase. During prophase, the complex of DNA and the proteins contained in the nucleus known as chromatin condenses. Now, the second stage, that is the metaphase. It lasts for only 2 to 10 minutes. It is a stage in the eukaryotic cell division in which the chromosomes align on the metaphase plate in the middle of the cells. As the microtubules are retracted, an equal tension is applied from each side of the cell chromosomes. This moves them to the middle of the cell. The chromosomes become arranged on the metaphase plate and are attached to the now fully formed spindle. Centrosomes are the opposite poles of the cell. So, in this picture you can see how the chromosomes are paired in the center. That means chromosome arranged in the metaphasic plate and attached with the spindles. Okay? So, the events which connect the chromosome to the spindle fibers and bring them to the metaphase plate are sometimes referred to as prometaphase. And the metaphase is the most suitable time to determine the number of chromosomes and study their morphology. Now, the third stage, that is the anaphase. It lasts for only 2 to 3 minutes. In this picture, you can see the sister chromatids of each chromosome separate from their respective mates and begins to move towards the opposite poles. It separates the duplicated genetic material which carried in the nucleus of a parent cell into two identical daughter cells. The separated chromosomes are then pulled by the spindle to the opposite poles of the cell. As the chromatids move apart, the polar fibers of the spindle elongates and the cells become longer. Here, the separation of chromatids in the picture you can see here, which starts at the kinetochores and the arms trail behind. So, the chromosome pulled into V, L, J or I-shaped depending upon the position of the kinetochores. Advantage of chromosome shortening is it is physically easier for a short compact chromosome to move through the cytoplasm than it is for a very long twisted interphase chromosome. So, the anaphase that ends when all the chromatids reach the opposite poles. And here, next is the telophase. It lasts for an hour or so. It separates the duplicated genetic material which is carried in the nucleus of a parent cell into two identical daughter cells. Now, the nuclear envelope slowly appears around each group of the chromosomes by coming together and the fusion of small vesicles formed in the prophase by breakdown of the parent cell's nuclear envelope and some portion of the endomembrane system. The chromosome gradually uncoil and become long and slender. The nucleoli start synthesizing ribosomes, the spindle fibers and the astral rays gradually become indistinct. Each pair of centrioles remains surrounded by the centrosphere, forming a centrosome. The centrioles may duplicate in the telophase in some cases. Viscosity of the cytoplasm decreases and the cell assumes in the normal forms. So these all are the functions and the process which occur in the telophase. Now in the picture you can see the non-disjunction in mitosis, which means the failure of homologous chromosomes or sister chromatids to separate properly during the cell division. So in this non-disjunction, it results into the daughter cells with normal chromosomes number. You see in one daughter cell trisomy, that means 2n plus 1 condition, and in the other that is monosomy, that is 2n minus 1 condition. Both cases are abnormal, and so one gamete has 3 chromosomes and the other has 1 chromosome. It causes disease which known as the chromosomal disorders. So in this picture you can see the mitosis is a type of cell division, the purpose of which is that two identical copies of a cell are formed, and the end result is that the DNA chromosomes replicate and one set of chromosomes with some of the cytoplasm and its content goes to each new daughter cells. So in this picture, first one is the interphase where the DNA is replicated, cell prepares for the mitosis. After that, it enters into the prophase where the chromatin condenses, making the chromosomes visible. Then it enters into the prometaphase where the nuclear envelope breaks down. Then the metaphase where the chromosomes become aligned at the equatorial plane. Then the anaphase where the sister chromatid separates and the resultant daughter chromosomes moves towards the pole. And the last is telophase where the daughter chromosomes reach the poles and form two nuclei. After that the cytokinesis occur and both the daughter cells separate from each other and form by a single zygote two daughter cells. Now after the karyokinesis, cytokinesis here, which is of two types, in which the first in the animal cytokinesis and second in the plant. You can see in the picture there is some differences in the animal cell and plant cell cytokinesis. In animal cell, cleavage furrow formed, but in the plant cell plate formed. So in animal cytokinesis, it occurs by cleavage. Here, the spindle starts disappearing in early telophase. Cleavage begins at the periphery and proceeds inward. Cleavage is started by contraction of the peripheral rings of the microfilaments. A midbody of the dense material is formed at the middle of the cell. As the furrow disappears, the midbody is compressed and becomes smaller and finally disappears as the two cells are fully formed. So this is the cytokinesis in case of the animals. Now the plant cytokinesis. In the picture you can see the comparison between the animal cytokinesis and the plant cytokinesis. In plant cytokinesis, cell plate forms which forms the middle lamella. After that the cell wall formation. So it occurs by the cell plate formation. A spindle persists till cytokinesis is nearly half through and it expands to form a barrel-like phragmoplast. Cell plates appear at the center and extends outward. Fusion of vesicles begins cell plate formation and the midbody is not formed. So here the cellulose, hemicellulose and the pectin are deposited by the cytoplasm of the daughter cells on either side of the middle lamella to form the primary cell wall. This divides the parent cell into two daughter cells. So it should be noted that the cytokinesis in an animal cell begins at the periphery and proceeds inward, whereas in the plant cell the cytokinesis starts centrally and proceeds outward. So this is the main huge difference between the plant cytokinesis and animal cytokinesis. In bacterial cytokinesis, the plasma membrane invaginates around the middle of the cell and new cell wall forms alongside. As the plasma membrane pinches, separates the closer towards the center of the cell, it's completely separate the daughter cells. And after this, the same process as the animal cell furrowing. Now, in this picture, you can measure the DNA content and the chromosome content in mitosis process. So here you can see the mitosis is a cyclic process, and here in the picture you see here the amount of DNA within a cell changes following each of the following events, like in fertilization, DNA synthesis, mitosis and meiosis. So we use here C to represent the DNA content, because content, the first letter is C in the cell, and the N to represent the number of complete set of chromosomes. Here the number, the first letter is N. In a gamete, the amount of DNA is 1C and the number of chromosome is 1N. Upon fertilization, both the DNA content and the number of chromosomes doubles to 2C and 2N respectively. Following DNA replication, the DNA content doubles again to 4C, but each pair of sister chromatids is still counted as a single chromosome. So, the number of chromosome remains unchanged at 2N. If the cell undergoes mitosis, each daughter cell will return to 2 carbon, 2C and 2N, because it will receive half of the DNA and one of each pair of sister chromatids. So in the picture you can see here 2C and 2N condition after the cytokinesis in the daughter cells, where it receives the half of the DNA, that is from 4C to 2C, and one of each pair of sister chromatids, that is 2N in number. Now the significance of mitosis. So, mitosis helps to maintain the size of the cell. A cell, when full grown, divides by mitosis instead of growing further. By mitosis, a fertilized egg develops into an embryo and finally into an adult by repeated mitotic divisions. It keeps the number of chromosomes equal in all the cells of an individual. It provides new cells to replace the old, worn out and dying cells. It produces new cells for the healing of the wounds and for regeneration. It brings about the multiplication in the acellular organism, and in the multicellular plays and role in the reproduction. Here asexual as well as sexual. And the last is it being essentially similar in many kinds of organisms, supports the basic relationship of all the living things. Now, the types of mitosis. Here, the first is intranuclear and extranuclear mitosis. In unicellular organisms such as amoeba and the yeast, mitotic events usually occur within the nuclear envelope, which remains intact, and the nucleus divides by furrowing along with the cytoplasm. It is known as the intracellular or premitosis, while the multicellular organism, the nuclear envelopes break down and the mitosis is termed extracellular or eumitosis. Second is anestral and amphiestral mitosis. In plants, there are no centrioles and no esters are formed. So, mitosis with ester is anestral, while in animals the esters are present, so mitosis is called amphiestral or estral. So, in case of the plants, because of absence of ester, it is known as anestral, and in case of animals, esters are present, so it is known as amphiestral. Next is endomitosis. In some kinds of cells, DNA and chromosomes replicate, but nuclei do not divide. This increases the number of chromosome sets per cell, called endoduplication or endopolyploidy or endomitosis. And the last is free nuclear division. In certain cases, nucleus divides repeatedly without the division of the cytoplasm, called free nuclear division. It produces multinucleate cells or syncytia. Here, Opelina, a protozoan, fungi, and endosperm cells of some seeded plants become multinucleate in this manner. So this is the complete types of mitosis in plants and animal both. Now, here the control of mitosis. Usually it is held that the surface volume of the ratio of a cell plays an important role. In this picture you see the spindle checkpoints, also known as metaphase to anaphase transition, the spindle assembly checkpoint. As the mitotic checkpoint, it is a cell cycle checkpoint during the mitosis or meiosis that prevents the separation of the duplicated chromosomes until each chromosome is properly attached to the spindle. So, mitotic checkpoint is in between metaphase to anaphase transition state. It has also been suggested that a change in nucleocytoplasmic or kern-plasm ratio in a growing cell or doubling of the DNA content in the interface may trigger the cell division. Cyclic changes in regulatory proteins are thought to function as a mitotic clock. The proteins are kinases which get activated by joining to another protein cyclin. The complex is called cyclin dependent kinases or CDKs, which we discussed earlier. And the uncontrolled mitosis leads to cancer. Now, the third cell cycle, that is the meiosis. August Weismann in 1887 predicted on the theoretical ground that the number of chromosomes must be reduced by 1/2 during the gamete formation. Edward von Benden demonstrated reduction division in 1887. J.B. Farmay and Murray in 1905 introduced the term mitosis. Only the cells of sexually producing organisms undergo meiosis and only the special cells in the multicellular organism switch over from the mitosis to meiosis at a specific time in the life cycle. So, the meiosis produces gametes in animals, some lower animals, and various protists and fungi. It also forms spores in higher plants. The complete meiosis process takes days to complete. The cells in which meiosis takes place are called meiocytes. Here, the meiocytes are of three types, in which the first is oocytes, which gives female gametes, that is ova or egg. Second is spermatocyte, which gives male gametes, that is spermatozoa or sperm. And third is sporocyte, which produces spores. Oocyte and spermatocytes are found in sex organs or gonads, and sporocytes are present in sporogonia. Now, the essential features of the meiosis. The two successive divisions without the DNA replication between them. Synapses of the homologous chromosome occur. Crossing over of the segments between the non-sister chromatids of the synapsed chromosome, and the segregation of homologous chromosome and the separation of sister chromatids. So these are the essential features of the meiosis. Now, the mechanism of meiosis. So, meiosis consists of two divisions that takes place in rapid succession, with the chromosomes replicating only once. Thus, a parent cell produces 4 daughter cells, each having half the number of chromosomes and half of the nuclear DNA amount present in the parent cell. So, it is known as reduction division. It is divided into two parts. First is first meiotic division or meiosis first, and second is second meiotic division or meiosis second. So, the first, that is meiosis first. In this phase, the two homologous chromosomes of each pair separate from each other and go to separate daughter cells. This reduces the number of chromosomes from diploid to haploid And so this stage is known as heterotypic division. So meiosis first is heterotypic division because here the number of chromosome reduces from diploid to haploid. It has 4 phases, in which the first one is prophase first. It takes more than 90% of the time required for the meiosis. It may be divided into 5 substages, in which the first one is leptotin. In this stage, a cell destined to become a gamete that enters the leptotin stage after its chromosomes are duplicated during interphase. During this stage, those duplicated chromosomes, each consisting of 2 sister chromatids, condense from diffuse chromatin into long, thin strands that are more visible within the nucleoplasm. Now the second, that is the zygote. The homologous chromosomes come to lie side by side in pairs called synapsis or syndesis. A pair of homologous chromosomes lying together is called bivalent. Pairing may occur in one of the following ways. So here in the picture you can see here the chromatids are present, which are known as bivalents, and after replication it forms sister chromatids or homologous chromosomes. So in pairing, the first one is the pro-terminal pairing. So the pro-terminal pairing starts at the end and proceeds towards the middle, while the procentric pairing begins at the centromere and progresses towards the end. And the last is random pairing. It commences at many points towards the end. Now the Pachytene stage. The chromatids of each homologous chromosome slightly separate and become visible, known as dyad. This stage is after the zygote, and a group of 4 homologous chromatids is called tetrad. The crossing over occurs during this stage. In this picture you see the 2 sister chromatids of the same chromosome are called sister chromatids, while the 2 homologous chromatids are known as the non-sister chromatids. So the chromatids of the same chromosome are the sister, while from the different chromosomes are non-sister chromatids. In this picture you can see the crossing over. Here, because of crossing over, you know that crossing over occurs in the Pachytene stage. Here the recombination involves mutual exchange of the corresponding segments of non-sister chromatids of homologous chromosomes. It takes place by the breakage and reunion of chromatin segments. This breakage is called nicking, which effected by enzymes endonuclease. Endonuclease assist both the nicking and the reunion called annealing, which aided by enzyme ligases. So the nicking, that is breakage, caused by the endonuclease, while the addition, that means annealing, occur by the ligase enzyme. So these recombinant chromatids after the crossing over forms non-recombinant chromosomes. Now the diplotin. The homologous chromosome starts separating called disjunction. Here the separation of homologous chromosome doesn't take place at the point called chiasmata. In the picture you can see the chiasma. The chiasmata indicates that crossing over is taking place. So on the chiasmata forms where the crossing over occurs. Next is the diakinesis. This stage is characterized by the shortening and thickening of the paired chromosomes, formation of the spindle fibers, disappearance of the nucleolus, and the degeneration of the nuclear membrane. So these are the characteristic features of the diakinesis. Now the metaphase first. It encompasses the alignment of the paired chromosome along the center of a cell, ensuring that the 2 complete copies of the chromosomes are present in the resulting 2 daughter cells of the meiosis first. Here in this picture, the attachment of tetrads to the spindle fibers in metaphase first is different from that of the mitotic metaphase chromosome. Here in the picture you can see each homologous chromosome has 2 kinetochores. one for each of its two chromatids. The two kinetochores of its homologous join the opposite spindle pole. So this is the metaphase in case of the meiosis. Now the anaphase first. It begins when two chromosomes of each bivalent separate and starts moving towards opposite poles of the cell as a result of the action of spindle. Notice that in anaphase first, the sister chromatids remain attached at their centromere and they move together towards the poles, which you can see in the picture. This is the stage of the meiosis anaphase first. Here the tetrads separate into individual dyads, that is chromosomes. Next is the telophase first. At each pole during this stage there is a complete haploid set of chromosomes, but each chromosome still has two sister chromatids. A cleavage furrow appears and by the end of this stage the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis. The daughter cells formed by the meiosis first are called secondary oocytes or secondary spermatocytes in case of females and male animals respectively. Now, what is the significance of meiosis first? It separates the homologous chromosomes. It introduces the variation by forming new gene combinations. It induces the cells to produce gametes or spores, and it may at times causes chromosomal mutations by abnormal disjunctions also. Now, in this picture, this is the stage of meiosis, which starts from a cell which has nucleus and chromosome from parents. This cell is zygote or fertilized egg. So after this, the chromosomes replicate prior to meiosis. Then like chromosomes start to pair up. Then the chromosome swap section of the DNA, that means crossing over, occur. After that, this creates a mix of new genetic material in the offspring's cells. Then it forms a two daughter cells because the nucleus divides twice into the daughter nuclei, and this daughter nuclei after the second meiosis forms the single chromosome and a new mix of genetic material. So in meiosis, in the first meiosis, two daughter cells forms, which after the meiosis second converted into the four daughter cells. Now meiosis second. In this division, the two chromatids of the each chromosome separate from each other and go to separate daughter cells. So, the number of chromosomes remains the same and it is known as homotypic division. It has four stages, in which the first is the prophase second. It occurs simultaneously in both the nuclei formed by the meiosis first. Centrioles already duplicated move apart in pairs. The chromosomes, each comprising two chromatids, become visible in the nucleus. They are set free in the cytoplasm. By the breakdown of the nuclear envelope, nucleoli disappear. Second is metaphase second. Here, the chromosomes takes up position at the equator of the spindle, forming a single metaphase plate. Then anaphase second, the two chromatids of each chromosome start moving away from each other and reach the poles of the spindle. Each pole has haploid number of chromosomes and haploid amount of DNA. Now, the telophase second. The group of chromosomes at each pole of the spindle gets enclosed by a nuclear envelope. Nucleoli are let down, estrol rays and spindle are lost. Now, after the telophase, cytokinesis occurs. Cytoplasm divides at its middle by furrowing in an animal cell and by cell plate formation in a plant cell. This produces two daughter cells. Here, the two daughter cells have half the number of chromosomes and half the amount of nuclear DNA. Meiosis second is not mitotic. Why? It occurs with haploid number of chromosomes. It is not preceded by an interphase. The daughter cells formed by meiosis second differ from each other, and the two chromatids of chromosomes are often not similar due to crossing over in meiosis first. Here, what is the necessity of meiosis second? You know that the aim of meiosis is to reduce the number of chromosomes to half. This is achieved by in meiosis first. So, what is then the necessity of meiosis second? Because the reduction division occur in meiosis first. So, what is the necessity of the meiosis second? Because the chromosome that separate in the anaphase of the meiosis first are still double. Each consists of two chromatids and have 2x amount of DNA. Thus, the reduction to haploidy in terms of DNA content doesn't occur in meiosis first. Truly, haploid nuclei in terms of DNA content as well as chromosome number are formed in meiosis second, when the chromatids of each chromosome are separated into different nuclei. Thus, the meiosis second is necessary. Now, in this picture, you can see how the chromosome segregates in meiosis first and second. In meiosis first, you can see at the prometaphase first, the homologous pair of chromosomes are held together at the chiasmata and the microtubules attached to the fused kinetochores of the sister chromatids. After the metaphase, in the anaphase stage, the homologous pairs of the chromosome are pulled apart by microtubules attached to the kinetochore, and the sister chromatids remain attached at the centromere. While in the meiosis second, in prometaphase second stage, the sister chromatids are held together at the centromere, while in the homologous pair of chromosome in the prometaphase first at the chiasmata. So, the microtubules attach to the individual kinetochores of the sister chromatids, and then in anaphase second, the sister chromatids are pulled apart by the microtubules attached to the kinetochores. So, in anaphase first, the homologous chromosomes pulled apart by the kinetochore in the microtubules, while in the sister chromatids are pulled apart by the microtubules attached to the kinetochores. Now, here you can see the differences in the mitosis and meiosis. So, in mitosis, it is a cyclic process, while the meiosis is not cyclic process. All mitotic divisions are alike, while the two meiotic divisions are dissimilar. First is reductional and the second is equational. In prophase is short and simple in mitosis, while prophase is long and comprising of 5 subphases in the meiosis. No chiasma forms or crossing over in the mitosis, while both occurs in case of meiosis. In mitosis, daughter cells have 2n amount of DNA, unlike the 4n amount in parent cell, while in meiosis, daughter cells have 1n amount of DNA, unlike the 4n amount of in the parent cell. Mitosis may occur in the haploid or diploid cells, while the meiosis always occur in diploid cells. In mitosis, chromosomes do not show chromomeres, while in the meiosis, chromosomes may show the chromomeres in case of meiosis. So these are the some differences in mitosis and meiosis process. Now, the types of meiosis. Here, the first, that is gametic or terminal meiosis. In this picture, you can see the gametic meiosis. It occurs in the protozoans, all animals and lower plants. Here, meiosis takes place before fertilization during the formation of gametes. The gamete formed by the meiosis are haploid. When the two gametes unite in fertilization, the resulting zygote is diploid. This type of life cycle with diploid adult and gametic meiosis is diplontic cycle. Second is zygotic or initial meiosis. In this picture you can see this. In fungi, certain protozoan groups and some algae, fertilization is immediately followed by meiosis in the zygote, and the resulting adult organisms are haploid. This type of life cycle with haploid adult and the zygotic meiosis is called haplontic cycle. And the third is sporogenetic or intermediate meiosis. In this picture, you can see the alternation of generation. In all the higher and some lower plants, gametes are formed by mitosis and meiosis occurs at the time of spore formation. Here, the spore may produces a multicellular haploid gametophytic phase which forms the gametes. Life cycle of plant is diplohaplontic because of having diploid and haploid multicellular phases. So these are the complete types of meiosis. Now, the significance of meiosis, in which the first is it produces gametes for the sexual reproduction. It produces gametes for the asexual reproduction in plants. It halves the chromosome number in gametes so that fertilization may restore the original diploid number in the zygote. It provides a changes for the formation of new combination of chromosomes which brings variations. Abnormalities in this process may causes chromosomal mutation. It begins essentially in all sexually producing organisms after further evidences of the basic relationship of the living organism. Now, the polyploidy. It increases in the number of full chromosome sets is called polyploidy. So, if the full set of chromosomes increase in number, then polyploidy may produce more vigorous offsprings, even new varieties and species. And the plant breeders have used colchicine induced polyploidy for producing new vigorous varieties of useful crops, fruit trees and the ornamental plants. Now, the cell division in prokaryotes. Cell division in prokaryotes is simple and doesn't occur by mitosis. It has two parts. First one is chromosomal division and the second is cytoplasmic division, which you can see in the picture in the process of complete cell division in the prokaryotes. At first, the replication of the circular prokaryotic chromosome begins at the origin of replication and continues in both directions at once. Then the cell begins to elongate. Here FTSZ proteins migrate towards the midpoint of the cell. Then the duplicated chromosomes separate and continues to move away from each other towards the opposite end of the cell. And here the FTSZ proteins forms a ring around the periphery of the midpoint between the chromosomes. Then the FTSZ rings direct the formation of a septum that divides the cell, and the plasma membrane and the cell wall materials accumulate. After the septum is complete, the cell pinches in two, forming two daughter cells. FTSZ is dispersed throughout the cytoplasm of the new cell. So, the whole process of replication and the cell fission is completed in about 20 minutes in rapidly growing bacteria. This is the end of cell division and the cell cycle process. Now, the next upcoming video is in the reproduction in organism. From this video, we will start CBSE class 12th topics in detail. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries, any suggestions, you can ask in the comment section below. Thank you for the watching.